Be going over E. coli and the lac operon. More specifically, we're going to be talking about gene expression. So let's say, for example, that you wanted to create a meal and you go into the library looking for information on how to create a specific meal. Now, would you go about reading every single book in the library? Well, of course not. What you're going to do is you're going to go to the cookbook section, you're going to grab a cookbook, and then look for your specific meal. So you might read 1 to 2 books, right? Well, just like you're going to read 1 to 2 books rather than read the entire library to figure out one thing, your genes are only going to want to turn on one at a time. And there's like a time and a place for each gene to be expressed. You don't want all your genes to be expressed at the same time because sometimes you might waste energy when you don't necessarily need to. So, for example, say we're getting a vitamin in the diet and you need, I don't know what gene this says, but let's say that this gene, when expressed, produces that vitamin within your body on its own. Well, if we're already getting that vitamin within the body, then you don't need this gene to be expressed because we're just going to waste energy by doing that. So what we do is we hold off on expressing that gene. So you may be thinking, well, how do we know whether or not, or how does our body function in a way to whether it represses some genes from being expressed or whether it allows other genes to be expressed? Well, that's exactly what this section is going to be talking about. So here we have gene regulation. So these vocab words here are going to be extremely important. We have our promoter. We have an operator, which is going to be stationed about right here. It's not in this picture, though. And then we have a start codon and a stop codon. And if you look carefully at like repetitive genes, this type of sequence is typical within human DNA. So here we have E. coli, okay? So before getting into E. coli and how it expresses its genes for a needed purpose, we're going to get into this word known as an operon. Now, an operon is a cluster of 3 genes that operate together. So in this instance, we have these 3 genes that when all 3 are turned on within an E. coli, what they do is they break down lactose. It expresses the genes that allow E. coli to break down lactose. Now, E. coli eats lactose. So if there is lactose in the area, we want these genes to be turned on, or the E. coli wants these genes to be turned on, I should say. But here we have that there are 4,288 genes in this E. coli bacteria. So how do we repress all the other genes while simultaneously expressing just a particular set of genes? So here we have what is called an operator, okay, or the ozone. Now, this operator right here, found there as well. This operator is the binding site for a repressor protein. A repressor protein. Now, what a repressor protein is it's a protein that latches onto the operator that halts the RNA polymerase from connecting to the promoter. Okay? So the repressor connects here, and that halts and gets in the way of RNA polymerase. Therefore, transcription cannot occur. Therefore, these genes cannot be expressed. However, if we have, in the example of E. coli, if there is E. coli, or if there is lactose floating around here, right? And then lactose, there's a little receptor right here where lactose can bind to. And when lactose binds to this receptor, this completely changes shape and pops off, which allows the RNA polymerase to connect to the promoter and finish up transcription of these 3 genes, which allows the E. coli to break down lactose for food. Now, this is going to be just another picture of it. It's just a different look at the same thing. Here's the operator, here's the repressor, and here's RNA polymerase being blocked. All right, and this is the other instance where the lactose comes, fits in with the protein. Protein changes shape, pops off the operator, and then we have this RNA polymerase that comes along and produces that mRNA strand to go out into the cytoplasm to produce its proteins through translation.